So, Dr. Wargo, since you're the newbie on Healthcare Unfiltered, I'd like to start with you and just a brief introduction about you.
Yeah, so my name's Jennifer Wargo. So I'm a professor of surgical oncology and genomic medicine at the University of Texas, MD Anderson Cancer Center. Been there about 10 years. Before that, I was at Harvard, where I began my studies on the microbiome, where we looked at the tumor microbiome in patients with pancreatic cancer. Since being at MD Anderson, I've started to study the gut microbiome and shown that gut microbes can actually help shape your immune system and can help shape cancer immunotherapy response. And then I've had the great honor and pleasure of working with others worldwide to look at other ways of manipulating gut microbes to improve response to cancer treatment, including with Monty and with Nazli, and super excited to continue that work together.
We're very happy to have you. The title of today's podcast, just so you know, the microbiome myths and facts, because sometimes you see so many things happening on social media and would like to make sure that listeners are aware of the true things and what's happening in that field, investigations, and then what are the things that are probably too much hyped that are not really necessarily true. Nazli, Dr. Dizman, I can say I've known Nazli before she was famous, you know what I mean? Like she's on her way to fame.
Famous.
No, but at some point she wasn't that famous, right? I mean, so then I'm gonna air the previous podcast when she was like one of us, and before I have to like, Nazli, welcome to the show again.
Well, you just wait. Give her a few years. She'll be uber famous, much more famous than any of us.
Trust me, I know.
You are all too kind. Thanks for having me here. Should I briefly introduce myself? Okay. I'm Nazli Dizman. I am a hematology oncology fellow at MD Anderson since the last 3 weeks. Prior to that, I was a resident at Yale, and prior to that, I was working with Dr. Monty at City of Hope as his postdoc fellow. My research interest areas are around the connection between the microbiome and outcomes in renal cell carcinoma and strategies to modulate gut microbiome.
Nazli, thank you for coming on the show. And Monty, one of the best mentors, honestly, I know in business. So welcome back to the show. Maybe a little bit about you.
Oh, sure. I'm a medical oncologist at City of Hope in Los Angeles. I've been here, gosh, for a long time now, 17 years at City of Hope. But, working with Nazli has been one of the greatest privileges, I think, of my career there. I'm so proud of her, and I'm incredibly grateful to Dr. Wargo for taking Nazli under her wing at MD Anderson. If it's okay, I'll just digress for 1 second and tell you a little story about Dr. Wargo, who I think the world of. On top of just being an amazing, accomplished scientist, this just shows you what a wonderful, collaborative human being is. She invited me to join a panel. I think it was at ESMO last year. And, she really did such a wonderful job of highlighting, everyone else's work. And at the end of the session gave me, this beautiful little plaque / trinket that I have on my desk. It was a globe, and, she'd written a little personal note in there saying, thank you for helping change the world of the microbiome. And I consider myself to be very new to the field. So I was just so touched by that, but it really speaks to her nature. So you picked a great array of folks for this podcast, Chadi, is what I meant to say.
And I think we all, for those of us who have had mentors that left an impact on us, you go to the grave thinking of these mentors, honestly. And I always say that there are people who think they are mentors just by simply if they help you with one thing or two things. But the reality is that it takes true person who's interested in you personally and professionally to really mentor you through a very difficult, the healthcare environment is not getting any easier, especially on the research side. But I want to start honestly with a very simple question. What in the world is the microbiome? Like what is like bacteria all over? So we're going to take the formalities away. We're going to be by first name, all of us. So Jennifer, we'll start with you. Like what are we talking about?
Yeah, it's, interesting tidbit. I always say that, 3.5 billion years ago, microbes actually helped to shape the earth for future forms of life, right? They actually gave us oxygen that we breathe today. And now 3.5 billion years later, we know that, not only is our environment filled with microbes, bacteria, viruses, fungi, you name it, but also our bodies are. And, in a particular individual, there's around 20,000 human genes, which represents 1% of the total genomic content, but there's between 2 and 20 million microbial genes, which represents 99% of the total genomic content. And importantly, it's inherently modifiable. Now, here's a piece of trivia for you, a cocktail party tidbit. So, there are more microbial genes in the human body than there are stars in the galaxy, which is super cool, right? Really great fact. And it goes without saying that because there's so many of these, that they can actually impact our physiology quite a bit. Sorry, I'm out with my kids. They're swimming here, so.
That's okay. That's healthcare unfiltered. We just do it all. Let me tell you, we don't filter
Sounds
anything.
good. But yeah, so these microbes, a lot of them reside in our gut. They have a profound impact on our physiology. They help us digest the food that we eat, and they also can actually help shape our immune system. If you think about it, all along that long alimentary tract, on one side you have these bugs, and on the other side you have these, this dense, infiltrating network of immune cells, this, mucosal associated lymphoid tissue or MALT. And so, really there's this constant and dynamic interaction between the microbes in the gut and the immune cells, and really an education of the immune cells.
But so Nazli, I mean, are we talking of the association of bacteria, viruses, fungi, parasites, and cancer? Is that what we're talking about? I mean, I want to simplify it even further. Are we talking about?
I think for the sake of this podcast, yes, I think limiting it to cancer is very reasonable, and saying that these microbes, so the microbiome refers to microbes and their genomes, right, within an area. And so, within a human, you have your human microbiome, and part of that is in the gut. And so I think, for the, really kind of to narrow the focus, I guess focusing on the gut microbiome in cancer and cancer immunotherapy response is probably the easiest to tackle in an hour or so.
Yeah, but in essence, that's what in essence, it is the association of, bacteria, viruses, fungi, parasites, and disease.
Yeah,
right?
And health for that matter.
And
they actually help keep us healthy, when we have the right balance in these bugs.
Nazli?
I was going to say, when we say microbiome, it again includes all those species, but you could also call more specific names for fungal microbiome, the fungal species under the microbiome umbrella as mycobiome, viral ones as virome, but in general, all those different microorganisms and all those different species, they would be called under the name of microbiome.
So Monty, what got you interested in this as a geomedical oncologist? Did you just start thinking that maybe some GU oncology diseases may be associated with microbiome, or?
It's actually a really bizarre story. And I don't know if I've really shared this with Jen before, but it was actually a biostatistician at City of Hope that got me interested in this. The biostatistician that I work with, and I've been working with for over a decade, has a lot of friends in the agriculture industry. And, as you can imagine, in the agriculture industry, modulation of the microbiome is everything. What you put into chicken feed and what you put into sort of livestock feed really has a dramatic impact in terms of the longevity of the animals, the extent to which they grow and develop and what have you. And of course, they've thought perhaps, far ahead of us about the benefits of fortification of the microbiome. So there's really sort of a rich science that's developed in the agriculture industry around this. He was motivated to sort of translate that into oncology. So, we had this paper out in CCR, I think it was in 2015 or something, 8 years ago, small paper, just sort of proof of principle, where we sequenced the gut in about 20 patients with kidney cancer. And all these patients at the time had gotten sort of the standard treatment back then, which was just a TKI alone, a VEGF TKI. And as we all know on this call, VEGF TKIs cause, unfortunately, a ton of diarrhea. So we were sort of using the microbiome to tease apart which bugs might be associated with diarrhea, which ones weren't. Tiny study, but that was my first foray into it. And then, I was really inspired by, Jen's paper, the work of others that came out, right around that time. really associating with checkpoint inhibitors, certain bacterial species, the diversity of the microbiome, all of those elements sort of came together. And I just started thinking, gosh, it'd be wonderful to, manipulate the microbiome as a means of sort of amplifying or augmenting the impact of checkpoint inhibitors.
So, Jen, do you want to take us back as to, what's the history that you saw something between microbiome and checkpoint inhibitors? Was this specific to a disease or just a class of drugs? And how is that link was established? What led to that?
Yeah, so I actually started to study the microbiome when I was, before I was at MD Anderson and when I was up at Mass General Hospital. And it was actually in the context of treating patients with pancreatic cancer. And what we found is that we were actually trying to test, what stromal factors might actually contribute to resistance to chemotherapy for pancreatic cancer. And we found that there was this one stromal cell line in our in vitro studies that was consistently able to confer resistance to treatment with chemo, across multiple different pancreatic cancer, colorectal cancer cell lines. And, we looked at different enzymes, which we thought it would be like in human tissues, and totally came up dry. And then on routine testing of the cell line in the laboratory, we found it was contaminated with mycoplasma, which at first was a bit devastating. But then we realized, well, wait a second, what if this mycoplasma is actually contributing to the therapeutic resistance? And we showed that if you treat the cells with antibiotics, kill the mycoplasma, you can actually, make the cancer cells sensitive again. If you infect another cell line, you can confer resistance and put it into a mouse model where we co-treated with antibiotics and chemo, and in the context of a colorectal cancer, and cured all the mice of their cancer. And so then went back to human tissues and found that, lo and behold, if we actually removed tumors of patients with pancreatic cancer and put them in an agar plate, you could actually grow bacteria out of those tumors. And, and then we published those findings in Science, and I think it was 2017, and I had been studying the tumor microbiome, moved down to MD Anderson and, went to a meeting actually and saw the great work of Laurent Zetvogel and Tom Gajewski, where they had showed in mouse models, depending on what the bugs look like in the gut, dictated whether or not those mice responded to cancer immunotherapy, and furthermore showed that you could change the microbiome and make the mice respond better. And so it was just one of those wow moments at a scientific meeting where I was like, this is amazing. I got up to the mic after the talk. I was like, this is incredible, you guys. Have you tested this in patients? And they hadn't. And so saw it as a unique opportunity to do that. And so ran back to MD Anderson, wrote a protocol to collect oral and gut microbiome samples in patients with melanoma who were going on to immunotherapy. In a year and a half, we collected samples from over 200 patients. Lo and behold, found that if patients had a more diverse gut microbiome, they did better. And if they had different bugs in their gut, they did better. A higher abundance of things like Faecalibacteria, Clostridiales, Ruminococcacea. And if they had a lower abundance of things like Bacteroides. And so we published those findings back to back with Tom Gajewski, who had a paper on patients with melanoma, and Laurent Zetvogel had a paper describing gut microbiome in patients with non-small cell lung cancer and kidney cancer. And so a trio of papers, the trilogy, if you will, was published in 2018 in Science, and then was really, very heartwarming to see a lot of scientists really, notice that and say, wow, we need to study this more. And, including Monty and Nazli and others. And now, gosh, it's like what they've done for the field far exceeds what we did, which is great. It's always nice to see people who
I think everybody recognizes the ones who started the field, frankly.
Yeah,
you're being too
village.
It does. Yeah. Obviously it does, but it clearly you started a wave and you need others to also help, and we're going to talk about that. Although I am curious, I want you all to comment when we get to it, as there are a lot of naysayers about the microbiome and say, like, I don't know what you're doing, and we'll go over that. Nazli, was that the work that started you and Monty thinking about how you can correlate this with kidney cancer? You're on mute.
Exactly.
Yeah.
That's the series of papers published in 2018 that kind of made us think that we could implement those studies in our cohorts of kidney cancer patients receiving targeted therapies and immunotherapies. We did two separate protocol, back to back, at times simultaneous, looking at the characteristics of the gut microbiome and the outcomes with different therapeutics in patients with kidney cancer. And then that these projects led to further projects trying to modulate the gut microbiome with live bacterial products.
So, Monty, was the choice of kidney cancer because you use IO? I mean, that's why you chose, I'm going to study the microbiome in kidney cancer because I'm using immunotherapy?
I think that it might have been more of a pragmatic choice because kidney cancer these days is pretty much all that I know. My clinic is, all about, 85 to 90% kidney cancer these days. I still love seeing my prostate cancer patients and bladder cancer patients, but, I will say that certainly, I saw the relevance within that domain. And, it's interesting. It's one of the things that I feel like happens less and less in this, post-COVID era. I mentioned the interactions that I had with my biostatistician over the, agriculture industry and, his interest there. What really sort of drew me to the sort of therapeutic domain of the microbiome were interactions that I had with our chair of immunology, Peter Lee, who's very involved in the development and distribution of CBM 588. Well, we can get into the details of that later, but it's a live bacterial product that Nazli and I have worked with over the past several years. And we would just have these water cooler conversations saying, hey, wouldn't it be cool if we, really sort of tested this particular agent in the context of immunotherapy? So, I do see the water cooler conversations coming back a bit. And that's definitely one of the peripheral benefits is just really, having those off the cuff conversations with colleagues and getting great ideas from just kind of hanging around.
Nazli, when you say I'm going to manipulate the microbiome, what does that even mean? How do you manipulate or modulate the microbiome?
Yeah, that's a great question. So as Dr. Wargo mentioned, several studies suggested certain species that are associated with better response to immunotherapies. So we hope that some type of intervention could modulate the gut microbiome, meaning that increase the abundances of those species associated with response, and perhaps that would impact a patient's benefit from the treatment. So that was our goal. That was also supported by the knowledge from 2 decades ago that the gut microbiome composition is impacted by one person's location, their occupation, their diet, the medications they use. So the gut microbiome is very dynamic. So we hope that an intervention that we picked would change the microbiome in a way that would support the immune system against cancer.
So, Jennifer, going back to some of right now, when there's a patient with cancer, I guess, I want to talk about healthy people as well, but I want to start with patients with cancer. Is the tradition right now, like if you're in the context of clinical trials, you collect stool specimens on everybody, blood specimen already, urine specimen, do we feel that there's, it seems like it's a lot because ultimately, while you're doing research, you want this to become pragmatic enough that it translates into clinical practice. Do you feel that eventually we are going to be in a scenario where stool is being sequenced for cancer patients and all other, other things?
Yeah, definitely. I think we do it in the context of clinical trials. I think we even do it on standard of care patients, but it doesn't, it costs something, right? But so do a lot of other tests that we do. We look at genetic profiling, we look at PD-L1, we look at tumor mutational burden. We look at next-gen sequencing, and all of these tests have a cost, but they also have a benefit. I think probably the hardest thing with the gut microbiome is that not everyone likes to collect poop, right? Patients and providers. And then B, that we don't know what a perfect microbiome really looks like, or at least historically we haven't. Now, I think we have a better sense, a more diverse microbiome is better. Why is that? It's because there's functional redundancy of these bugs. And it's not necessarily the names of the bugs that matter, but their function. But for many years, the signatures that we saw in patients with different types of cancer who responded to immunotherapy treatment, depending on where those studies were done and what type of cancer, it was different, right? And so you couldn't say, there's the perfect microbiome, right? But I think now there have been larger cohorts of patients studied in aggregate, and they actually do find that there's some unifying signatures. So, the short answer is yes. Will we be collecting and sequencing more poop samples from patients? Absolutely. And I think now with advances in next generation sequencing, we can do that faster, better, and we can also have more clarity about what a good microbiome really is, and then what to do with that information, right? Because that's the key.
Yeah, I think, Monty, I think the reason I asked the question is because while nobody wants to collect poop, but if you're going to get information that you can get elsewhere, people will go through the trouble of it. A lot of the folks who are skeptical about microbiome research, they say, okay, it's good research, but it's not adding much. I can get the other information somewhere. And even if I'm getting information, it's not really altering what I'm doing, things of that nature. So as a scientist, when you approach this, you're looking at the pragmatic piece. You're trying to think, okay, how this is going to translate pragmatically. And B, how do you answer the question that this is really not giving you additional information beyond what you have already?
Yeah, I think all of us sort of grew up in this genomics age where, we would actually, the sort of experiment and dabble with these various platforms, across disease types. And I really liken it to that. I think the way that we really figured out how to utilize genomic data for our patients, and I'm referring, of course, to DNA and RNA sequencing of the tumor, is just by doing it in sort of broad scope in large series across multiple histologies, and then really sort of linking and correlating that to various treatment outcomes. And voila, now we have this age where in a lot of fields we're actually using precision oncology. I love Jen's approach of really trying to collect stool specimens on as many patients as possible, doing the sequencing. I certainly concede that we're not there yet, but I have no doubt that we're going to get there when it comes to being able to link a certain stool bacteriomic profile, perhaps with benefit or lack thereof with therapy. And I think, moreover, I can really envision an age now where we have tailored therapeutics directed at the microbiome, and I think it holds that certainly the stool microbial profile would be a very logical, predictor of benefit. I mean, all of this is hypothetical, but nonetheless, I think likely, in the sort of time that we'll all be practicing as oncologists.
Nazli, you've done some research on kidney cancer, and you've published some of this work with Monty and others. But can you use this as an example so at least listeners understand the type of research that you did? And I think there was I'm forgetting. I forgot the title of the entire paper, but I think you know the paper I'm talking about.
Yes, that's a very long title. So we started a project with Monty around 2017, 2018, enrolling patients with metastatic renal cell carcinoma prior to starting first-line treatment with nivolumab and ipilimumab. And we randomized patients into either receiving nivo-ipi alone or receiving nivo-ipi with CBM-588, which is a live bacterial product developed in Japan in the 1960s and have been used in Japan very commonly, mainly in GI conditions for so many decades. So in the study, we looked at several outcomes. The primary endpoint was the increase in the bifidobacterium species, and the secondary endpoints were the clinical outcomes, such as progression-free survival, response rate. Although the sample size was small, we had a total of 30 patients, and 10 of them were randomized into nivo-ipi arm, and then 20 were randomized into nivo-ipi and CBM-588 arm. We have seen some interesting findings. The study didn't meet its primary endpoint, so bifidobacterium didn't change from baseline to week 12 in either arm. But when we looked at the patients who had benefit from treatment with CBM-588 in addition to nivo-ipi, we saw an increase in bifidobacterium species, which may suggest that some patients actually benefit from the additional live bacterial product on immunotherapy combination. And these patients also achieve a gut microbiome modulation. In addition to that, I think the most interesting findings were in the clinical data. Although, again, it was a small data set, the progression-free survival with nivo-ipi CBM-588 at the first analysis was like 12 months. Sorry, that's my dog. He thinks that I'm talking with him. In the progression-free survival in the nivo-ipi arm was 2.6 months. That was a big difference. And response rate was 20% versus 58%. So these were the findings that really attracted the attention of like several clinical and translational researchers and scientists. We also have found supportive data showing increase in chemokines in patients who received CBM-588. And, several other findings in gut microbiome that may support the use of CBM-588. This compound was later tested in a very similar design in patients who received nivolumab and cabozantinib. And this was recently presented at ASCO as an oral presentation. And the findings were pretty much in line with what we found in the past.
Jennifer, there are a lot of people who, congratulations. There was a lot of hypothesis generating in this. Obviously, it's not really changing anything today, but certainly setting the stage for a lot of things moving forward. It makes me wonder, there's a lot of research on diet and cancer. And in fact, I'm airing an episode with Dr. Urvi Shah, who's done a lot of work on myeloma and diet, in a few weeks. But I wonder, is there a link there? Like, the type of diet that leads to a particular microbiome, that leads to a particular, is that how the link is? And then what do you say to people who say that probiotics, if I take probiotics, this is something really good for prevention of cancer now in healthy people?
Yeah, great questions. I think, so there's a couple different ways you can modulate gut microbes, right? You can actually give a fecal transplant. And there were 2 back-to-back papers published in Science showing that if you give patients with metastatic melanoma who progressed on anti-PD-1, a fecal transplant from either a patient who had a complete response or a partial response to treatment with immunotherapy, you can actually reverse immunotherapy resistance. Really spectacular studies. But again, fecal transplants are difficult, right? And finding cancer patients.
I'm not going to lie, they sound a little bit awful, fecal transplant.
Yeah, it's not so bad, though. When you're facing a diagnosis, it's not so bad. You know what I'm saying?
Yeah,
but,
I do.
I can tell you that, we ran a study and we tried to get cancer patients to be donors, and we really couldn't because they were all excluded, right? And so then just last week, there was a really beautiful paper published in Nature Medicine showing that in patients with melanoma, frontline therapy with fecal transplant from a healthy donor is actually safe and effective. It was a single arm study, so hard to really compare it, but nonetheless, you can even use a healthy donor. Now the important thing though, and so for the longest time, we're like, okay, we should just use fecal transplants, which is a stepping stone. It's not the end game. And, until, we saw Monty and Nazli's work, we were thinking, well, gosh, it's just going to have to be FMT for now. But wow, to see, a live bacterial product like CBM-588, essentially a probiotic, be effective in treating cancer alongside treatment with immunotherapy was just spectacular. Now, how about diet? And so this ties back to really to the function of these microbes. It's not just their names, it's what they're doing, that really matters. And
You're on
so,
mute. Yeah, you went on mute. Okay.
oh yeah, sorry, I went on mute just to
No
make
problem.
sure my kids are safe in the pools.
Yes, safety first.
Yes. So, but anyway, it's all about function, right? And you can change the function of microbes, a couple of ways. One is by diet. So it's not just, who those bugs are, it's what you're feeding them that matters, seed and soil, right? And so we showed in a publication in 2021 that if patients eat a high fiber diet, they're much more likely to respond to treatment. Okay. And so for every 5-gram increase in fiber in their diet per day, they actually had a 30% reduction in the risk of recurrence. Really dramatic, right? And so we took that back to a mouse model, gave mice a low fiber diet. They failed to respond to treatment, and they failed to activate T cell signaling. Hey, quiet guys. And then we have, now we're running dietary intervention studies with Jen McQuaid and Carrie Daniel, where we actually take patients with melanoma and other cancers and give them high fiber diet.
Yeah, that is, I mean, that to me, it's fascinating because, I think everybody, I mean, there are two issues from patients with cancer, and Monty, maybe you'll reflect on that. Patients with cancer, while it is difficult with dietary manipulation, but they will always prefer that over chemotherapy. Let's be honest here. And then for healthy people, if you tell them, if you take probiotics once or twice a day, and if you do a couple of things, you reduce your risk of developing cancer, it will be a very welcome way of doing things. So I guess my question to you now, with your observation with Nazli's paper and with what Jennifer said, how are you taking that, and what are the type of next generation studies that you're thinking about in that particular area?
Oh, can I add one more thing though before we leave the diet? In that paper, we actually showed that if patients reported taking commercially available probiotics, that they actually numerically did worse. And this was, kind of common probiotics in the US. Yeah, not CBM 588, not Japanese probiotics, but American probiotics, And then we took it back to a mouse model and actually gave mice, germ-free mice, a perfect microbiome from a complete responder donor, and then treated them with either sterile water as a control versus one of 2 probiotics we bought off the shelf, either Culturelle or Align, Bifido or Lactobacillus, right? And if then we implanted tumors and treated with checkpoint blockade, and what we found is that if mice received either of the commercially available probiotics, these 2, either Bifido or Lactobacillus, that they actually did worse, and they had worse immune systems. So not all probiotics are good. So
very
I guess
interesting.
They all want to know what they can do to stack the deck in their favor, right?
Yeah. So Monty, as a researcher, you're designing a lot of trials, obviously focusing on the GU world a little bit. Are you starting to add a lot of this as secondary endpoints? Are you trying to add a component in some of the interventional studies, collecting stool, adding some other interventions? How are you taking all of this and applying it to the kidney cancer world, which you live in day in and day out?
So I assume you're referring specifically to diet, probiotic intake, and so forth.
Diet and probiotics and microbiome and things like that.
Certainly things we're trying to capture. I don't think anybody's doing it better though than Jen's group. But if you don't mind, I'll turn it back to you, Jen, because I think that it would be wonderful to hear a little bit more about the prospective fiber diet study you guys are doing. I just think it's so cool.
Let's hear it, Jen.
Yeah, so we have these dietary intervention studies where we'll give patients up to 50 grams per day of fiber. Now, it's not fiber you can buy in a pill. It's actually whole food based fiber, with fruits and grains and veggies and, we actually have a chef, chefs that we've hired, and we have a bionutrition research core, essentially a food kitchen. We actually package meals, kind of like Blue Apron, and ship them anywhere in the country, right? And we've started these dietary intervention studies. The diets are very tolerable. We can get patients up to 50 grams per day, which is really important. And we're seeing some encouraging responses, though, results are early.
Are you doing this for, I may have missed it, in melanoma specifically or in other cancers?
Now we're doing it in multiple cancer types. So
Okay.
all different cancer types. And now this is data that, like these are guidelines you can give patients too, right? Like having adequate dietary fiber of at least 20 grams per day is good advice to give to patients, right? And something that, we don't necessarily, need to package, nor can we package meals for every single patient, and ship them anywhere in the world. I think we can do education efforts and really enable patients, to eat the right things.
So Monty, I want to go back to you because I think these dietary interventions are great. How do you, I guess the question is from a scientific standpoint, how do you know that this is directly contributing? Because there's so many other things happening, the confounding factors that may be leading to the outcome that you are measuring. Sometimes could be the additional therapy that you are giving to the cancer patient, and patients get different treatments. Could be the host microenvironment in particular cancers. I guess my question is, how much can we tell that the particular dietary intervention is indeed making the difference versus it is associated to the outcome, and it's part of the confounding factors that we may be dealing with?
Yeah, I think that, it's so critical that we just, kind of maybe trust randomization in these cases. I know it's hard to do, right? The studies we're doing, at this point in time aren't definitive. We've got to get there. But, I think if we look at the data and the context of the randomization, at least we can see whether or not there's a signal there to move forward with. That's why I love, sort of what Jen's group is doing with the dietaries. I believe there's a randomized component to that, if I remember correctly, which is
I may have missed that.
Critical.
Yeah, I see.
Yes,
I may have missed that.
Them to an isocaloric diet that's not high fiber. You know what I mean? So either same number of calories, high fiber versus, around 15 to 20 grams per day.
Jen, is there a specific cancer in your mind that you think is more prone to the microbiome manipulation? Or obviously you've done a lot of work in melanoma and other cancers, but is there, do you think colorectal cancer, just because it's the colon and small bowel and these, like this would be more prone to manipulation, or it really doesn't matter because melanoma has nothing to do with I don't know. I'm asking an immature question, but I promise you it's a question that's coming through the mind of my listeners.
I love this question. And, it's a great question. So there was a paper published out of Memorial Sloan Kettering, right, in Nature Medicine that showed in CAR T therapy, so chimeric antigen receptors, right, treating liquid tumors, that if patients got antibiotics and wiped out their gut microbiome, that they actually didn't respond and they had higher toxicity, right? And now they are actually giving things like fecal transplants, to patients who have liquid tumors who are getting stem cell transplant or CAR T, to really kind of make their microbiome better. And if you think about it, like in a lot of these solid tumors, what do patients see before they see immunotherapy? Chemo. And, what does chemo do to your microbiome? And it can be quite rough on the gut microbiome. So, melanoma, it's a little easier to study just because we can give frontline immunotherapy, but I still think the microbiome has relevance in every cancer, the gut microbiome in particular.
Yeah. So Nazli or Monty, or both of you, what are the, put yourself in the position of a reviewer who is reviewing a paper talking about microbiome. What would you say the shortcomings of microbiome research? What are the issues that you hear around you of folks who might be skeptical, I guess? And I believe sometimes skepticism actually helps because it leads to more research and more definitive conclusions. But at least, the skeptics about this type of research, what are they skeptical about? What are the things you hear them saying, and how do we respond to them? Nazli, maybe I'll start with you and then Monty can comment on that.
Yeah, I can go. So I think there are several of them. I think earlier in the mid 20-teens, so the sample sizes were similar. I think that was perhaps a limitation that was cited a lot. And at that time and up until now, causality is another component of limitations that mentioned in many studies. It's in line with what you mentioned, is that they were going to respond, then their microbiome changed, or their microbiome changed, and because then they responded. But nowadays, like most of the researchers like us, like try to work with the lab-based scientists, like try to replicate the findings and laboratory animals to kind of observe there is causality actually, as we speculate that. Aside from that, I think for years the microbiome analysis have evolved. Now we are going beyond just calculating the number of the or percentage of the specific species. Now that we're doing more transcriptomics, we are looking at the functional gut microbiome and combining all those like different data points. So that is an improvement that has been cited as a limitation.
You went on mute, Nazli. I think you
I
went
did.
on mute.
Oh, sorry. So I was gonna the last thing.
Until you repair your audio, I'm gonna ask Monty something. You weren't on mute. Monty, you can hear me? Monty?
Yeah. Can you hear me okay, Shadi?
Yeah, I can hear you. I was gonna say,
okay.
you're wearing your swag shirt. So I have to ask you a question, because that's actually important. I mean, I would think that the cooperative groups would be so well positioned to answer a lot of questions if you guys are collecting stool specimens, frankly, because you run these large studies that change standard of care in certain scenarios. And I could imagine a situation where you can go back to some of the prospective studies and analyze the specimens and see if you can get some information. Are you doing that right now? Is this in the future? I mean, are we at a stage where part of any cooperative groups, you may not sequence right away because there's funding issues, but are you saying collect the specimens and store them and see what we do with them later on?
So, I'll tell you that it's not quite yet within the GU group, although we have some discussions underway. But, at our last SWOG meeting in April, we had some terrific talks from the group focused on checkpoint inhibitor toxicity led by Nicole Kuderer. And they have a project ongoing right now within SWOG called I Check It, a brilliant name to really sort of capture prospectively IO-related toxicity. And within that study, I believe they're already actually doing stool collections. So, that was a really sort of interesting insight into what's happening at that cooperative group level. Within the GU space, we're having some discussions around moving CBM forward into a trial that really builds nicely on Nazli's design. It's at very early stages of concept discussion. I feel pretty comfortable sharing it, though. Pedro Barada, who's at Case Western, brilliant young GU investigator, is actually leading the charge with that trial. So I'm hoping that, SWOG and other cooperative groups will play a very active role in further examining these compounds.
Well, I'd like to finish by each one maybe saying concluding statement. I can tell you, I'm very excited watching the field. I'm obviously not actively involved in this, but I'm really very, I feel there's something there. I believe we, at some point, you all are going to find something. My concern is not the science, to be honest. My concern is more the pragmatic thing. Are we able to translate the science eventually into practicality of what people do? Like Jen said, nobody likes to collect poop, not the patient nor the physician. So maybe concluding remarks, we'll start with Jen.
Yeah, I think, and I think there was some hesitation early on to collect these samples. I think people are excited about it now. Even in venues where I'm meeting people out, we talk about the microbiome. People want their poop sequence now. So I think there's a lot less reluctance, and knowledge is power. And you can do 23andMe and with your blood, and you can do 23andMe for your poop, right? And so who wouldn't want to know, especially since it's actionable, right? You can, if you know what's there, how you can optimally change it, right? So I think with regard to profiling the microbiome, it's going to become more frequent. Will we have better ways of doing it, that are more user-friendly? They have these smart toilets that they're developing, believe it or not, where you can sit down and it'll sample your microbiome and then send back information to your smartphone. So, someday could we actually have a smart toilet that kind of tells us what our gut health is like? I think that day is in the future. I think also they have these 3D printed pills that you can swallow that'll sample your gut microbiome along the length. And then they have these breathalyzers that measure volatile organic compounds, which are kind of the output of the gut microbiome. And so could that be a way to profile? And then I think from an intervention standpoint, it's moved so rapidly already. I mean, the first studies were done in mice, published in 2015. The first human cohort studies, 2018. And then the first human intervention studies, 2021 with FMT. And then, wow, CBM 588, now the sky's the limit, right? And so now there's a lot of groups trying different ways to change the gut microbes, right? Through either live bacterial products, through FMT, through dietary intervention. We're actually looking at targeted antibiotic approaches that selectively deplete bad bugs and let the good bugs grow, and CRISPR, can you actually engineer either someone's inherent microbiome or a new microbiome? We've created these synthetic communities of microbes that could actually be given to patients or to healthy individuals to optimize their microbiome. So I think the future's bright. It's going to be a very pillar of not only cancer care, but overall precision health. Ultimately, we just need to just accept it and adopt it.
That's amazing, Jen. Thank you. I wrote a book on glyphosate and non-Hodgkin lymphoma and Roundup. So I have a lot of interest in the impact of pesticides on gut, but I don't want to digress about that. So I was an expert witness against Monsanto in the first 3 trials. Nazli, your final comments.
It's always so hard to speak after Dr. Wargo, after being that much inspired with that. So maybe I'll take my few minutes to thank our patients. I echo what Dr. Wargo said so far. We collected over almost 150 samples, of which 60 were from interventional studies. And our patients really dedicated their time. They collected their diet every day. They brought their stool sample at the beginning, like through FedEx in huge boxes. Now it's like pretty small and very convenient. But, they really helped us kind of just improve ourselves along the way. And I really appreciate their participation and their help to us to help understand, the impact of gut microbiome and how we can modulate it.
Thank you. Monty, last comments?
Yeah, tough to go after these two, but what I'll tell you is that, I feel like so much of what we do on oncology, right, is dictated by, biotech. And, I think that there may be some initial stumbles that cast some doubt on the field. You think about, Ceres Therapeutics, you think about 4D Pharma and so forth. But, those are just two, sort of singular approaches towards modulating the microbiome. You've heard from Jen about all the amazing things that they're doing to manipulate the microbiome at MD Anderson. As I look around the globe, there's so many diverse approaches, beyond CBM, beyond FMT, which, by the way, comes in many different flavors. I would tell you that there's other approaches, building consortia of bacteria and administering those to patients as opposed to singular strains, which is what, we've sort of done today. Tons of exciting approaches that are coming in the future. And I definitely think that, I'll echo something I said earlier, hopefully within the time that we're in practice, we're going to see some of this really become a standard of care. I definitely think with bright minds like, Jen and Nazli and others, we're definitely destined to do that.
Well, I'm very grateful. Yeah.
Can I add one more thing? Antibiotic stewardship, I think, is really important. So they have actually shown, David Pinato and others, Bertrand Rowdy, have shown that if patients get broad spectrum antibiotics before they get treated with immunotherapy, they fail to respond, right? And so, we really need to be careful about who we give these broad spectrum antibiotics to. And does it have an impact on overall health? I think that's something else. Like you said, pesticides, quality of drinking water, broad use of antibiotics. Is this contributing to the development of cancer in younger individuals? I think these are important questions that we need to study.
I couldn't be more thankful, Dr. Wargo, Dizman, and Pal. Thank you so much for coming on Healthcare Unfiltered. I definitely need to have you again next year. There's really I view this is an educational opportunity to a lot of folks. So, with several thousand people listening to this podcast every week, I think hopefully your voice will reach folks who probably don't know about this research from patients, families, and students and fellows and others. So I couldn't be more thankful. Thank you so much for coming on.